it's me Erica Moore and welcome back to my YouTube channel. So this video is the part 2 of the discussion of protein synthesis, and for this part we are going to talk about the translation. For starters, the instructions for making proteins are written in a cell's DNA, and it is in the form of gene. Basically, a gene is used to build a protein in a two-step process. Step 1. Transcription. Here, the DNA sequence of a gene is rewritten in the form of RNA. In eukaryotes, like you and me, the RNA is processed to make the final product called a messenger RNA or mRNA. Step 2. Translation. In this stage, the mRNA is decoded to build a protein that contains a specific series of amino acids. Requirements for translation. The process of translation or protein synthesis involves the decoding of an mRNA message into a polypeptide product. Amino acids are covalently strung together by interlinking peptide bonds. Each individual amino acid has an amino group and a carboxyl group. Polypeptides are formed when the amino group of one amino acid forms an amide bond with the carboxyl group of another amino acid. This reaction is catalyzed by ribosomes and generates one water molecule. The protein synthesis machinery. In addition to the mRNA template, many molecules and macromolecules contribute to the process of translation. Translation requires the input of an mRNA template, ribosomes, tRNAs, and various enzymatic factors. Ribosomes. A ribosome is a complex macromolecule composed of structural and catalytic rRNAs and many distinct polypeptides. Ribosomes exist in the cytoplasm in prokaryotes and in the cytoplasm and endoplasmic reticulum in eukaryotes. Ribosomes are made up of two subunits. In E. coli, the small subunit is described as 30S and the large subunit is 50S for a total of 70S. Mammalian ribosomes have a small 40S subunit and a large 60S subunit for a total of 80S. The small subunit is responsible for binding the mRNA template, whereas the large subunit sequentially binds tRNAs. tRNAs. The tRNAs are structural RNA molecules that were transcribed from genes by RNA polymerase III, serving as adaptors. Specific tRNAs bind to sequences on the mRNA template and add the corresponding amino acid to the polypeptide chain. Therefore, tRNAs are the molecules that actually translate the language of RNA into the language of proteins. Mature tRNAs take on a three-dimensional structure through intramolecular hydrogen bonding to position the amino acid binding site at one end and the anticodon at the other end. The anticodon is a 3-nucleotide sequence in a tRNA that interacts with an mRNA codon through complementary base pairing. tRNAs need to interact with three factors. First, they must be recognized by the correct amino acid synthetase. Second, they must be recognized by ribosomes. Third, they must bind to the correct sequence in mRNA. Amino acid tRNA synthesis through the process of tRNA charging, each tRNA molecule is linked to its correct amino acid by a group of enzymes called amino acyl tRNA synthetases. At least one type of amino acyl tRNA synthetase exists for each of the 20 amino acids. Steps of translation. As with mRNA synthesis, protein synthesis can be divided into three phases: initiation, elongation, and termination. The process of translation is similar in prokaryotes and eukaryotes. Initiation of translation. Protein synthesis begins with the formation of an initiation complex. In E. coli, this complex involves the small 30S ribosome, the mRNA template, initiation factors, and a special initiator tRNA. The initiator tRNA interacts with the start codon AUG. Guanosine triphosphate (GTP), which is a purine nucleotide triphosphate, acts as an energy source during translation, both at the start of elongation and during the ribosome's translocation. Once the appropriate AUG is identified, the 50S subunit binds to the complex of methRNAi, mRNA, and the 30S subunit. This step completes the initiation of translation. Elongation of translation. The 50S ribosomal subunit of E. coli consists of three compartments. The A site binds incoming charge amino acyl tRNAs. The P site binds charged tRNAs carrying amino acids that have formed peptide bonds with the growing polypeptide chain but have not yet dissociated from their corresponding tRNA. The E site releases dissociated tRNAs so that they can be recharged with free amino acids. This creates an initiation complex with the free A site ready to accept the tRNA corresponding to the first codon after the AUG. The formation of each peptide bond is catalyzed by peptidyl transferase, an RNA-based enzyme that is integrated into the 50S ribosomal subunit. The energy for each peptide bond formation is derived from GTP hydrolysis, which is catalyzed by a separate elongation factor. The amino acid bound to the P site tRNA is also linked to the growing polypeptide chain. As the ribosome steps across the mRNA, the former P site tRNA enters the E site, detaches from the amino acid, and is expelled. Amazingly, the E. coli translation apparatus takes only 0.05 seconds to add each amino acid, meaning that a 200 amino acid protein can be translated in just 10 seconds. Termination of translation. Termination of translation occurs when a nonsense codon UAA, UAG, or UGA is encountered. Upon aligning with the A site, these nonsense codons are recognized by release factors in prokaryotes and eukaryotes that instruct peptidyl transferase to add a water molecule to the carboxyl end of the P site amino acid. This reaction forces the P site amino acid to detach from its tRNA, and the newly made protein is released. The small and large ribosomal subunits dissociate from the mRNA and from each other. They are recruited almost immediately into another translation initiation complex. After many ribosomes have completed translation, the mRNA is degraded so the nucleotides can be reused in another transcription reactions. That's the end of the discussion for protein synthesis. So I hope you learned something about our topic, and protein synthesis is a process in which cells make proteins, and it has two stages, the transcription and translation. Thank you for watching and see you on my next video.